Okay, today's anatomy topic is cells, and we're going to focus on cell division, cancer, and tonicity. So let's get right to it. Cell division. What is the cell cycle actually? Well, it's a sequence of growth and division in cells. The cell cycle consists of some main parts. There's interphase. Interphase is like sort of like the rest period, but it's not very restful because what happens is the cell is doing its regular cell job. So if it's a muscle cell, it's contracting. If it's a skin cell, it's protecting the outside of the body. Okay, if it is a bone cell, it is performing its function. So no matter what kind of cell it is performing its cellular function. But also near the end of interphase, it's getting ready for division. So it's going to copy organelles and copy the DNA. Then we have the mitotic phase, that's division. And then we have cytokinesis, which is the splitting or dividing of cells at the end. Okay. At the end of the cell cycle, so we complete the whole thing, interphase, mitosis, cytokinesis, we have two identical daughter cells, genetically the same. What are the functions of cell division? Well, cells divide for growth, repair, replacement, and reproduction. So grow, it's how you get bigger, get more cells, obviously more cells than when you were born. Repair, so if you have a wound or something is damaged. Replacement, so cells are going to age, so we need to replace those. And then for reproduction, so meiosis, but also unicellular organisms are going to reproduce that way. So what actually is mitosis? Mitosis is division of the nucleus. Remember, the DNA is found inside the nucleus. And mitosis is a process that, because it divides the nucleus, it keeps the number of chromosomes the same, which is critical for all cells having the DNA needed to function correctly. So here's a visual of the cell cycle. You can see most of the cell cycle is spent in interphase, growing, copying the DNA, preparing for division, copying organelles. Okay, but in interphase, remember, it's performing its normal cell life. In fact, all but about maybe 24 hours is going to be in interphase. So if a cell lives for 3 months, most of the time it's going to be spent in interphase. Okay. So what would happen if cells were always dividing? So that's a critical thing to think about as we talk about this next piece. Okay, what stage in the cell cycle is not part of mitosis? Well, that's interphase. Remember, we talked about it. It's when cells grow and do their regular cell job. So what would happen if cells were always dividing? They skip that long period of interphase. Okay, so imagine a cell starts this size, but it doesn't grow, it just divides. And then those cells don't grow, they just divide. They don't grow, they just divide. So if all cells did was divide and skipped interphase, the size of the cell would get smaller. They would have less and less of the DNA, less and less of the materials. Because remember, the DNA is copied in interphase. So if we're skipping interphase, we're shrinking the amount of DNA. And so those cells really are not going to be functional. And we talked about the end of the cell cycle being cytokinesis. So what is that? That's when the cytoplasm gets divided and we actually get those two daughter cells created. And then what is apoptosis? Well, apoptosis, P.S., one of my favorite words, but it is programmed cell death. It's a process that helps to eliminate cells with mistakes. So how do cells even know there are mistakes? Well, there are checkpoints. And there are points where the cell examines some internal and some external cues and decides whether or not to move forward with division. And if there's a mistake, there's a problem, the cell may be pushed through apoptosis to prevent a problem from getting through. Okay, so it's like self-checking. Okay, so it's like imagine an assembly line, oh, this one's missing something, pull it up. Okay, that's really what's happening with checkpoints and then pushing that cell toward apoptosis to keep that defective cell from dividing and passing it on to further cells. However, it's not a perfect process. So what happens if mistakes in division do get through? it can lead to cancer, and cancer is uncontrolled cell growth. So what are some causes of cancer? So we have carcinogens, environmental factors that can lead to the development of cancer. So things like radiation, things like chemicals that are in tobacco smoke, okay, or in vape juice. It could be pollution from factories, okay. A lot of things can be carcinogens. It could be a mutation, so just simply a mistake. So my cell was dividing, it missed getting caught by the checkpoint. However, only about 5 to 10% of cancers actually result from a random mutation that happens where we have a mistake in the code. And there is a family history or genetics link. It can make you more susceptible to cancer, but it's not a guarantee. Okay, however, there is a hereditary component to a lot of cancers. And what are the stages of cancer? So we're going to start with stage 0 and go to stage 4. Stage 4 being the most serious, where the cancer has metastasized. Metastasized means it has spread from its original locations. Say, for example, it was stomach cancer and it spread somewhere else, to your lymph nodes, to your lungs, okay? So as you move from stage 0 to stage 4, it's more serious, harder to treat. Okay, because once it has metastasized, more organs are involved. Okay, now we're on to the topic of tonicity. So you may have learned this before, but there are 3 key terms. So there's isotonic. Isotonic is when the solute and water concentrations inside and outside the cell are equal. So what happens is water moves into the cell, water moves out of the cell, but it happens at the same rate. So it's going in and out, in and out, in and out, in and out. So what happens as a result is the cell stays the same size. So isotonic does not mean that water is not moving. It just means that it's moving in and moving out at the same rate. Then we have hypotonic. Hypotonic is when the solute concentration outside the cell is lower. The water concentration outside the cell is higher. So sometimes we talk about it in terms of solutes, and sometimes we talk about it in terms of water. Okay, so hypotonic means there's lots of water outside the cell, but very little solute, okay, or at least less. So what happens is, since there's so much water outside, water comes in much faster than it's leaving. So here's water leaving and water coming in. Okay, it's coming in much faster. So what happens is the cell starts to swell up and it can burst, and the word for that is lyse, L-Y-S-E. Then we have hypertonic. Hypertonic is when the solute concentration outside the cell is higher and water is lower. So less water outside the cell, more water inside. So what happens this time is water moves out of the cell much faster and moves in very slowly. So out fast, in slow, out fast, in slow. So what happens in this case is the cell shrinks and shrivels, and that's called crenate. Okay. So here's an illustration of what that would look like when we're looking at red blood cells. Okay, again, in hypertonic, you can clearly see from the picture, water is moving out much faster. The cell shrinks or crenates. Isotonic, we've got water moving in, water moving out, same rate, cell stays the same size. Hypotonic, water is moving in much faster than it's moving out, and the cell can swell and eventually burst or lyse. So why is this important? Why are we talking about this in terms of cells? Well, remember osmosis, diffusion of water. So diffusion is when things move from high concentration to low concentration. That happens with osmosis, but osmosis by definition is when that happens with water. Okay, so osmosis is diffusion, but specifically it's diffusion of water. So cells will lose or gain water depending on the environment. And since we're made of cells, it's happening in our body. So the water concentration, the environment that cells are exposed to determines whether or not they're going to lose and gain water. So if you drink salt water exclusively, okay, which is why it's bad if you were like stranded on the ocean or something. So drinking salt water, it makes your blood very hypertonic, and your cells are going to be shrinking. You're going to get dehydrated. Even though you're drinking, you're still going to get dehydrated. And distilled water is going to make if you drink distilled water exclusively, again, it's going to be problematic because your cells are going to be swelling and potentially bursting. So we have to think about this in terms of IVs. When someone gets an IV, they have to be given an isotonic solution, meaning the same concentration as what blood normally is in terms of what solutes are available. So if you look at IV fluid bags, it'll say saline. It's not pure water. It has solutes in it. And then probably the most interesting thing about this, for me anyway, is related to diabetes. When your blood sugar is high, this means it creates a hypertonic solution in your blood, and your blood's flowing past cells, contacting them. And this causes it causes cells, not just your blood cells, but other cells that it comes in contact with, but especially red blood cells, to lose water. So those cells start shrinking. This loss of water by those cells is recognized by the body as dehydration, and you start to feel thirsty. So thirst is one of those common symptoms or warning signs for diabetes, and it's because the high blood sugar creates a hypertonic solution in the blood. So there's a fun fact how it relates to actual real life. So yes, tonicity is important in real life. All right, there we have it. Now you've learned about cells, at least all you need to know for me.